Living organisms can be classified into two groups, prokaryotes and eukaryotes. The major difference between the two groups is that prokaryotes lack membrane-bound organelles like the nucleus, mitochondria, endoplasmic reticulum, etc. Whereas eukaryotes are organisms that contain nucleus and other membrane-bound organelles inside the cell. In the case of prokaryotes, once a gene is transcribed into an mRNA, its translation into a protein starts even before transcription is completed. So if a gene is transcribed, it means that the gene is expressed into proteins. But this is not the case with eukaryotes. Having a compartment like the nucleus for the storage of the genetic material allows eukaryotes to have an additional level of gene regulation. This means that having a gene transcribed into an mRNA in the nucleus does not guarantee its translation into proteins. Only if the transcribed mRNA is transported to the cytoplasm will the mRNA get translated. This allows the eukaryotic cell to store mRNAs in the nucleus if the translated protein is not required immediately. So in order to export eukaryotic mRNA out of the nucleus, it has to undergo three processes collectively known as the post-transcriptional modification. The first modification made to the mRNA is capping. This involves adding a specially altered nucleotide to the 5' end of the RNA. This takes place co-transcriptionally, which means that the modification is made to the mRNA even before its transcription is completed. Capping involves three enzymes: RNA triphosphatase, RNA guanyl transferase, and guanine N7-methyl transferase. First, RNA triphosphatase removes the 5' terminal gamma phosphate from the 5' triphosphate of the RNA. Then, RNA guanyl transferase consumes a GTP molecule and adds a GMP group to the 5' diphosphate in an inverted orientation, causing the two nucleosides to be joined by an unusual 5' to 5' triphosphate bridge. Finally, the guanine N7-methyl transferase adds a methyl group to the N7 amine to form the 7-methyl guanosine cap. This cap serves several functions. It binds to proteins that aids in the transport of mRNA out of the nucleus. It prevents the 5' end of the mRNA from being digested by exonucleases, and it recruits proteins called initiation factors, which are necessary for mRNA translation. The next modification is called tailing, and it determines the 3' end of the mRNA. Usually, transcription proceeds around 1000 bases beyond the actual site that will become the 3' end of the final mRNA. Around this site is a conserved sequence called the polyadenylation signal, which is recognized by an endonuclease that cleave around 20 nucleotides downstream, and then an enzyme called poly-A polymerase adds around 250 adenosines one after another without any template to form the poly-A tail. The poly-A tail plays a crucial role in mRNA stability. It recruits poly-A binding proteins, which prevents the exonucleases from accessing the ends of the mRNA. This is important because exonuclease can only remove nucleotides from either the 5' end or the 3' end of an RNA molecule. These proteins also help improve translation efficiency by interacting with the eukaryotic initiation factors bound to the 5' cap. This causes the mRNA to adopt a circular shape which allows ribosomes to easily reinitiate translation after completing a round of protein synthesis, making the process more efficient. The third modification is called splicing. In this process, non-coding regions are removed from the RNA transcript so that protein coding regions can be joined together to form the functional RNA. These regions that are expressed into proteins are called exons, while the non-coding regions inside a gene are called introns. Eukaryotic genes contain introns because it allows different combination of exons to be included in the final mRNA, leading to the production of a family of related proteins from a single gene. The process of splicing is mediated by a large RNA protein complex called the spliceosome. It binds to conserved regions at the boundaries between an intron and exon and acts as an enzyme removing the introns and then joining the exons. Certain chemical factors can cause the spliceosome to act differently causing different combinations of exons to be joined together, and this is what allows multiple proteins to be made from a single gene. Once all three modifications have been made, the mRNA is exported out of the nucleus into the cytoplasm, where it is used as a template to synthesize proteins.